Imagine a microscopic organism, something that feels like it's straight out of a Marvel movie, acting as a tiny genetic spy.
Right.
It swims through, say, your body, finds one specific dangerous strand of DNA, and then reports back what it found.
And the thing is, this isn't science fiction anymore.
Not at all. This is real engineered bacteria, and they're being trained to detect the genetic signatures of, well, everything from cancer to pollution.
Today, we're doing a deep dive into this exact topic, looking at a really comprehensive review article.
Yep, it's titled Bacteria as Living Biosensors for DNA, and it was just published in Nature Reviews Bioengineering.
The authors are a team primarily from the University of California, San Diego: Catherine O'Connor, Paige Stepp, Daniel Worthley, Jeff Hasty, and Robert Cooper. They've basically laid out the whole field of what they call bactosensors.
And our mission today is to unpack how these living bacteria are engineered to be such sophisticated biosensors. We're talking about precision that's almost hard to believe.
Yeah, being able to spot a difference of just a single base pair, it's incredible. And this isn't just a cool science project, right? This is about changing where and how DNA testing can even happen.
Okay, so what's the core problem they're trying to solve here? Why do we need this?
Well, it really comes down to accessibility and cost. Right now, our gold standard for DNA detection is PCR.
Which is amazing,
But it requires these huge, expensive machines. You need sterile, climate-controlled labs, highly trained staff. It's not something you can just do anywhere.
So the goal with bactosensors is to create something low cost, something you could use at the point of care or, out in a field somewhere.
Exactly. Something that needs minimal equipment and almost no sample processing. It's a huge shift in thinking.
Instead of building a billion-dollar machine to analyze the biology.
Yeah, you're using the biology as the machine.
Oh.
We're capitalizing on something bacteria already do naturally, the superpower they have to just suck up foreign DNA from their environment.
So we aren't adding a whole new function. We're just reprogramming the reporting system.
That's it. We're telling it what to do after the DNA is already inside.
Okay, so let's get into the blueprint. How do you build one of these things? I know any biosensor basically have three parts, right? A sensor, a converter, and a reporter.
Yep. Sense it, transduce it, report it. And with a bactosensor, the living cell does all three jobs.
And it starts with picking the right vessel, the living chassis.
Right. And the review points to a couple of star players. One is Acinetobacter bailey. It's what's called naturally competent.
Meaning it's just always ready to eat environmental DNA.
Exactly. It's always on the lookout. Another big one is Bacillus subtilis, which you can engineer to become super competent.
So how do they actually grab the DNA? It feels like it would be just floating around.
It's this amazing physical mechanism. The bacteria has something called an uptake pilus.
Okay.
Think of it like a tiny molecular grappling hook. It extends out, captures free-floating DNA, and reels it back towards the cell.
Wow.
But it doesn't just swallow the whole thing. The selectivity starts right at the front door.
What do you mean?
There's a channel in the membrane called COMEC. It's like a very, very picky bouncer at a club. It only lets a single strand of the DNA double helix come inside.
Ah, okay. So it unzips it on the way in.
Precisely,
Yeah.
which is critical for the next step.
So the DNA is inside. What stops the bacteria from just, chopping it up for food? Isn't that what usually happens with foreign DNA?
That's where the engineering comes in, the map, so to speak. Once that single strand is inside, the cell uses a natural process called homologous recombination.
Right.
And we, the engineers, have built what's called a landing pad into the bacterium's own genome.
And this landing pad is designed to recognize the specific DNA we're looking for.
The landing pad is flanked by what are called homology arms, and those little arms are designed to perfectly match our target DNA, say a specific mutation from a cancer cell.
So if the DNA from the environment matches the arms,
The cell's own repair machinery gets tricked. It sees a perfect match and it performs a swap. It literally cuts out the old sequence on the landing pad and pastes in the new one.
it's like a molecular handshake that confirms the target is there.
And that handshake is what triggers the whole detection circuit.
But wait, if you're working in a really messy environment, like intestinal contents or soil, how can you be sure the right DNA makes that swap? Isn't there just a ton of random genetic noise?
That's a great question, and it speaks to the robustness of this. The cell's machinery already prefers a precise match. But more importantly, you don't need every single cell to make the swap. You just need enough of them to recombine to generate a signal.
So it's a numbers game.
It is, and the data shows it works surprisingly well. The review cites work, including from Cooper and his colleagues in 2023, where they successfully identified DNA from pathogens and human tumor cells in completely unpurified samples.
Unpurified, that's the key word.
That's the game changer. You don't need a sterile lab and a purification kit. You just need the sample, maybe a little heat, and the engineered cells.
Okay, so that's general detection. But what about getting super precise, like telling the difference between a harmless gene and a harmful mutation? That often comes down to a single letter change, a SNP,
And that's where you bring in the brains of the operation, the CRISPR-Cas system.
the famous genetic scissors.
We usually think of them as an editing tool, but here they're used for targeting. Remember, CRISPR is originally the bacteria's immune system.
Right. It stores a memory of enemy DNA.
So we repurpose that system to give the bactosensor that laser-like specificity. It's what allows it to tell the difference between two sequences that vary by just one single base pair. It's like the targeting computer on an X-wing.
So once the correct DNA recombines, how does the cell, raise the alarm? How does it create a signal we can actually read?
I love how clever this part is. The review lays out 3 main strategies, and they're all based on a really smart principle: deletion. The successful recombination event deletes something that was holding the output back.
It's like cutting a tripwire.
Exactly. Let's say the output you want is for the cell to glow green. The first, and maybe most versatile strategy, is deletion of a repressor.
Okay, so a repressor is a protein that's actively holding the glow green gene in the off position.
Correct. The landing pad is placed right where the repressor gene is. So when the target DNA recombines, it kicks out the repressor gene. No more repressor protein gets made. The block is removed.
And the light turns on.
The light turns on. The second is deletion of a terminator. This is more like removing a physical roadblock on the DNA highway.
A stop sign for the machinery that reads the genes.
Perfect analogy. When recombination happens, that stop sign sequence gets deleted. So the machinery just keeps on going down the line until it hits the output gene and expresses it.
And the third one sounds dramatic. Deletion of a toxin.
This one is very high stakes. The output here isn't light, it's survival.
So if the bacteria doesn't find the target DNA,
The toxin gene remains intact, it gets expressed, and the cell basically self-destructs.
so only the cells that successfully detect the target DNA survive to tell the tale.
It's a powerful selection tool. It ensures that what you see growing at the end are only the successful sensors.
That's brilliant. Okay, let's get back to that wow fact, this single-base detection. What was that specific example with the KRAS gene?
Right. So Cooper's team engineered Acinetobacter bailey with its own native CRISPR system to spot that single base mutation in the KRAS gene, which is a major red flag for colorectal cancer.
And they tested this in a living system,
They
right?
did. They showed these engineered bacteria could actually detect tumor DNA that was just spontaneously shed into a mouse's gut. It was non-invasive, highly specific detection happening in vivo.
Inside a living, complex, messy gut. That's
Mhm.
incredible. And it takes this idea beyond just detection, right, into this realm of theranostic.
Yes, which sounds like something straight out of Star Trek, I know.
It really does.
It's the fusion of therapeutics and diagnostics. A theranostic biosensor doesn't just raise a flag when it sees a problem, it acts on it.
So the bactosensor could detect the cancer DNA.
And then as its output, it could release a localized therapeutic, maybe an anti-inflammatory molecule or something to trigger the immune system.
So you get incredibly precise, localized drug delivery right where the problem is.
Avoiding all the side effects of flooding the whole body with a drug. It's truly personalized, location-aware medicine.
So if we compare this to other CRISPR systems like Sherlock and Detector, which are already in use for things like COVID tests, why is this approach better?
Well, those in vitro systems are powerful, but they are still lab-bound. They need purified samples, precise temperatures, expensive machines to pre-amplify the
You
DNA.
still need the clean room, basically.
You do. Bactosensors leapfrog that whole bottleneck. Because the bacteria do the work themselves, they can process these unpurified samples. It just drastically cuts down on the complexity and the cost.
Which makes them perfect for resource-limited settings. You mentioned the World Health Organization has criteria for this kind of thing.
They do. It's the reassured criteria. It's an acronym for affordable, sensitive, specific, user-friendly, rapid, robust, equipment-free, and deliverable.
And bactosensors tick a lot of those boxes.
They hit the biggest ones for field use. They're affordable, they're robust, and most importantly, they are almost entirely equipment-free. The living cell is the equipment.
And the applications go way beyond medicine.
Oh, absolutely. Think about agriculture. You could deploy these to detect available nutrients in the soil or, pollutants like heavy metals in a river.
Using bacteria to report on the local chemistry of an environment they already live in, it makes perfect sense.
But this all leads us to the single most important question.
Safety. Biocontainment. If you're releasing engineered organisms into the environment or into a person, you have to be absolutely certain they don't stick around forever or share their new genes.
It's completely non-negotiable, and the review covers several layers of really ingenious safety strategies that are being built.
Okay, walk us through them. How do you keep the microscopic spies from going rogue?
First up is attenuation. This is basically just making the bacteria less fit to survive in the wild. You might delete a gene that's essential for its growth, like the perm gene in Salmonella.
And without that gene?
It can only grow in very specific purine-rich environments, like a tumor. If it leaves that target area, it essentially starves.
Smart. What's next?
Kill switches. These are engineered genetic circuits. Think of them as remote self-destruct buttons. They're often called dead man or passcode switches.
So the cell needs a constant signal from you to stay alive.
Exactly. You provide a specific molecule. If that molecule disappears, or if it encounters a different kill signal, the circuit triggers cell death within hours.
Then there's physical containment, right?
Right, physical encapsulation. You can embed the bacteria in a kind of porous hydrogel. Small molecules like the target DNA can get in, but the bacteria themselves are too big to get out. It's like a molecular cage.
But the gold standard, the one that sounds most foolproof, is genomic recoding. That sounds intense.
It is profound. This involves systematically going through the bacteria's entire genome and replacing certain genetic words or codons with a different version.
So you're changing the language it speaks.
You're making it dependent on a synthetic amino acid, a building block of protein that you have to supply externally, one that doesn't exist anywhere in nature.
So if it escapes, it can't build proteins, it can't grow, it just stops.
It stops. And because its genetic code is now fundamentally different from every other microbe on the planet, it can't pass its engineered genes onto wild bacteria.
That's effective.
Incredibly. They've achieved undetectable escape frequencies in tests with up to 10 to the power of 11 cells. It's the level of safety you'd need for widespread deployment.
Okay, so let's tie this all together. What are the big takeaways for you from this deep dive into the work of O'Connor, Cooper, and their colleagues?
I think there are three big ones. First, just that living bacteria are being engineered with tools like CRISPR to detect DNA with a specificity we've just never had in a portable system before. The cell is the detector.
Right. Second takeaway. Bactosensors thrive where traditional lab tests can't go, in complex, unpurified samples with minimal equipment. This is a huge deal for global health.
And finally, the really exciting frontier is theranostic. Not just diagnosing diseases like cancer, but using these same living detectors to simultaneously deliver a targeted, localized therapy.
It really shifts the entire paradigm. We move from expensive centralized labs that react to disease to these low-cost, deployable systems that can proactively monitor and treat.
It blurs the line between diagnosis and therapy.
So, a final thought for everyone to chew on. If we can program bacteria to safely detect and treat disease inside the body, what other hard-to-reach, difficult-to-monitor environments could these microscopic spies unlock for us next?
You could be talking deep-sea vents, monitoring ecosystems on a mission to Mars, early warning systems for crop disease. The potential for analysis in the field is it's limitless.
A fantastic thought to mull over. Thank you for joining us for this deep dive into the world of living DNA biosensors. We hope you feel a little more well-informed.
And if you enjoyed this look at complex research made simple, please make sure you subscribe to the deep dive for more breakthroughs you really need to know about.